The best garden in the street. Don't forget the sea salt.
Hi, Nigel Ruck here. Now, you all know I love my gardening, and we all know that sea salt is simply fantastic for keeping your garden healthy and looking great. But did you know this? Sea salt reduces transplant shock and dramatically increases a plant's survival rate when it's just been planted. It's like insurance for your plants. So when you're putting new plants in your garden, don't just water your plants, sea salt your plants.
Don't forget the sea salt.
We are absolutely thrilled to have a traditional Australian company like Seasol sponsoring us here at All the Dirt.
Hello and welcome to All the Dirt podcast. I'm Darren Thorpe. Today's guest is botanist Professor Kingsley Dixon, a former WA Scientist of the Year who's made many discoveries that have transformed the way we think about plants and conservation. Kingsley spent 32 years as the Foundation Director of Science at Kings Park Botanic Garden, and he got the glamour gig promoting WA plants in David Attenborough's BBC series The Private Life of Plants. Today he works as a professor at Curtin University, building collaborative teams from different disciplines to undertake the challenges of restoring Australia's landscapes. So, hello Kingsley. So, let's go right back to the beginning because that's what we always do. Tell me about growing up in Beachborough.
So Beachborough was a real experience. We were on an acreage. It was a little house built out of cement bricks that my parents had made. And actually we had the foundations right next door, limestone blocks for the grand house, but they were always just limestone blocks.
It never happened.
It never, ever happened. Raised on rainwater from our windmill. And the great joy for me was, as a little kid, seeing this thing called the bush. I had a mother who was a complete snake phobe. Mm-hmm. Being embedded in complete Banksia woodland, of course, there were snakes. And that's probably the thing that stayed with me most of my life is that snake phobia. But as a child, I remember walking in the bush, and I can recall it in Technicolor, and I can remember, which I now know as herbaceous, I can remember the hovea, which was sparkling, and I can remember the summer starflower with their extraordinary lipstick pink flowers. And they were just welded in. So there was this thing where I was being infected by our wildflowers, so that for the rest of my life, I'm the happiest walking in a piece of bushland.
When you left school, you studied botany.
That was my ambition. 14th of February 1966 was the big epic moment in my life when we went to decimal currency and I saw the $5 note with Joseph Banks on one side. Mm-hmm. And he was a person who had the title of a botanist, and I realised you could actually become a botanist. Now, at that stage I was just 12 years old, but that's where I knew what I wanted to do. And at my school at that stage, I went to the vocational guidance officer. That meant you went on to a job. And I said, I really want to become a botanist. And she said, oh, yes, I've checked that all up. And, yeah, you join the Faculty of Agriculture.
Oh, that's...
Which wasn't the fit because I discovered at UWA a building with the word botany on the front. And that's when I went and discovered there was a whole department. And I did my undergrad, I did my honours degree straight through to my PhD. And it was just a joy. It was hard, but it was a joy.
So, so you did that at UWA, University of Western Australia. You also taught there. But one of the things I admire about you is you're not just an academic botanist. Like, I always think of botanists, they sort of, look at plants and they categorise them and they work out how it, all comes together and how they work in the environment. But you're really practical. For instance, when I first got into gardening, which was a very long time ago, we always thought that seeds germinated from a fire because of the heat. But you undertook research that showed it was something else that stimulated germination.
Well, being raised in the bush and often driving around with my parents, we always loved picnics up in the Darling Range because that was exotic. There were things like Leschenoltia up there, which we didn't get down in the sand plains. We had Leschenoltias, but they were very dull little things. And we'd go to spots in spring when there'd been a previous summer fire because the wildflowers were always better. So I was always hooked on this whole thing. And when I started my research career at Kings Park, one of the very first and early projects that we were approached with was by a company that said, we really want to propagate this rare and endangered beard heath. It's a member of the native blueberry family, a thing called leucopogon, now called Stiphelia. And I said, oh, yeah, that's fine. We'll do that. We put the seeds in, and as everybody knows, for these hard-fruited ones, we get about 5% up, 95% of the viable seed just sits there and says, we're not germinating. And then we started the same work that we realised everybody else had done for 150 years, way back to the British stove houses when early seed was being sent from Western Australia to Britain. And we were doing heat and ash because we thought it comes up after a bushfire, it has to be heat and ash and heat and ash and heat, and we did that. Lots of experiments, lots of research scientists in Australia publishing on heat and ash and the minor effects it has. And then I had the great joy of listening to an obscure talk at a botanical meeting in, of all places, Rio de Janeiro. And hardly anyone turned up to this talk, but I was drawn to it. A novel application of smoke for germinating South African species. And I went along to the talk, and there was just something that sort of settled. There were no questions. It was a 15-minute talk. It finished, and we all left. But I recall so vividly on the flight home, because I dropped into Cape Town and visited their labs by coincidence. So I went and saw their trials. But on the way back to Perth, I just didn't sleep because I kept thinking, of course, the third thing from a fire. How could we, for essentially 150 years, have ignored the third thing? Now, it took us a year to work out how to do it because the South African method didn't work. And then one day it all happened at Kings Park, and we had amazing things. We had yellow bells, we had callitrix, we even had Eastern States flannel flowers just coming up like hairs on their cat's back. And it was just one of those extraordinary, and it was truly a eureka moment because it transformed so much. So all of the conostylus, all the little cottontails you see along freeways, they're all grown with smoke. And I'm thinking that was a little contribution, and that's great to see.
That's a great contribution. Your team named the smoke molecules carrageenanoides. Is that right?
Well,
or
that's very Italian. Carrageenanide.
Yeah. I did study Italian at one time.
Yeah.
So it means carrick means smoke in the local Noongar language. Is
That's
that
correct,
right?
yeah. And olide because it's part of a class of molecules called butanolides. So what you do is you then pick up the latter part of that word and put carrageenanide. So we're really, really thrilled to have honoured our Noongar culture with that name.
So obviously you can't always, when you're doing it, set up a smoking, and then you develop something called smoke water.
So one of the things that we were interested in is how can you package this so that people don't have to set up a little smoking machine. And sure, people have had bee smokers and Weber kettles and garden sheds and old car bodies. They've had everything imaginable. And so we thought there was a great opportunity for the Friends of Kings Park at their wildflower sale that we could actually have little bottles of smoke water, because we were using smoke water. It wasn't as good as what we call the aerosol smoke that you create from putting it into a tent and smoking your seed. But we made the smoke water, and I brought a bottle along, and
I'm going to sniff it now.
Yeah, and it's like bushfire in a bottle.
It is like bushfire in a bottle.
Isn't it extraordinary?
The colour of it is sort of a pale amber colour, and that's from the
But the great
burning
thing with this is we're now training a couple of local nurseries to make their own smoke water so that people can germinate all those wonderful things. And it's about a 30%, just over a third of West Australian plants absolutely adore being soaked in smoke water and then so on. And generating the smoke water the way we're training some of these nurseries means you get the highest quality. And so free of charge, I've developed a quality assurance. We have a local weed that finds germinating in the smoke water irresistible. And so they can send me their batch and I can say, you have a 4-star smoke water, you have a 5-star smoke water, because we can calibrate it against our standard.
So that's just had benefits, for agriculture, for, restorative work we do
Colossal.
looking after, land care work and horticulture. But you also proved it wasn't just Australian plants that are smoke germinated.
No, that was the real eye-opener, and it came from colleagues in Scotland who burn the heather. Now, they burn it because they go grouse shooting, but they said if they don't do that, they don't get germination. So we got some Kaluna seed, which is the genus, and sure enough, you get a bit germinating without smoke, but you get it all germinating when you add smoke. So I'm going, wow, so it's there. And then we started checking species from non-fire prone habitats. Certain rainforest species with small seeds also respond. And so there's a more generic basis to this, even in non-fire prone environments, that we think the carotenoid molecule and some of its sister molecules in the class of carotenoids, that they also get stimulated by being in the soil seed bank, and then an animal might dig that rainforest soil. And because you get tillage, you potentially get what we call an oxidative burst. The microbes go nuts, and you may get a little pulse of the carotenoid molecules because it's just oxidation of organics that make our chemical. Just like burning, it's very rapid oxidation. So nature's been able to join the dots in a range of different ecosystems through this common molecule.
So are they using your smoke water all around the world?
Yep. The molecule, carotenoid, is the newest, what we call phytohormone, plant hormone. So it's shown to be a generic molecule that does lots of other things in plants, including inside the plant. It helps regulate branch length and branching patterns. So it has a basis in real plant architecture. So plants that you see use carotenoid to signal within themselves, and they've worked out when a seed germinates, it's actually a miniature plant being told it's time to start growing this point and branching there. So the molecule just does that, but it does it at extraordinarily low levels. It's about a teaspoon in a normal domestic swimming pool.
Wow.
And that gives you the same strength as the smoke water we generate through our smoke machine.
Plants are just, the more you learn about them, the just more fascinating they become.
Yes, they are fabulous.
Now, another one of your discoveries was devising a method of seed treatment called flame ablation. So how did you get the idea for this one?
So it came from the fact that we wanted to begin restoring native grasses. We have a lot of native grasses. We have some particularly very good native grasses in the southwest. They're deep-rooted, they're perennial, not like the nasty rain-fed annual grasses where we're now finding with shifting climate that these annual grasses don't do so well, whereas the deep-rooted natives do. The natives also resist heavy grazing. They hold the soil together. But the problem is they have seeds with all sorts of bits on them. So you can't put them through an automatic seeder. They jam up the seeder. So how do we clean them up? How do we make them look like a wheat grain? Because a wheat grain is a very sophisticated structure where they've taken all the bits off. So you see that. But most grass seeds have all these appendages and hairs. And then there's lots of other species with fluffy things. So it means putting them through automated seeders to put them on landscape scale is not going to work. And that made us think that maybe there's a method to do this. And the crazy idea came to me, and it was from looking at one of these advertisements for one of these hamburger companies, and they showed hamburgers going across a flame grill to get the charring, but without essentially doing much more than charring. And I thought, I wonder if we could adapt it. And so working with some colleagues and a very gifted engineering student, and this was our first foray into stepping outside the botanical comfort zone and into the nasty world of the engineers. And he came along, we looked at the machinery, he started looking into temperatures of flames, and then we got a drum, rotating drum, and you could spin the drum, adjust the flame intensity, and we could get perfect burning off for many species of those nasty appendages. And that was flame ablation.
That's just, so that means that you can have thousands of seeds rather than...
That's right.
Before that, people were having to hand clean the seed, I presume.
Really hard, and lots of health issues with it because of all of those fine hairs. Those silica hairs can cause allergies. Now we've moved the technology on even further, and now we're about to make some announcements about the way that we've been able to put these little seeds into a smart seed coat. So we're creating new synthetic coats around seeds that give them all the instructions when to germinate, how to germinate, the insecticides and all sorts of things targeted exactly where it matters at the emerging plant. So it means farmers no longer have to broad acre spray. The little seed will come with its own package of special information, including the carotenoid, which is the first time we've managed to deliver it in one of these seed pellets, these seed coats.
That is absolutely amazing because, we always thought that we were getting high tech when we put polymer coats on our fertiliser to do the slow release fertiliser. And this is a lot more sophisticated than that.
And a lot harder, but it will transform the restoration of Western Australia's and Australia's landscapes, because in this way, our plan is to take many native seeds and to put them through a canola seeder.
Mm-hmm.
So a canola seeder, a farmer can sow 1,000 acres a day. Currently, the seeders we have for native seeds are tiny little things. They're like overgrown garden machines. And so our vision is to get many native seeds bulked up to the size of a canola seed with their smart coat, which means every seed matters, which means we will use potentially up to 90% less seed because we'll be more effective at our seeding. So it's a space to watch over the next year.
Wow. That's just amazing. Your other collaborative work has been in orchid biology and ecology. You've looked at, hormones and all sorts of things. So tell us some of the interesting things. I'm thinking about discussing about how female wasps attract males, because I think that I find that particularly fascinating. The fact that plants are so clever.
Well, everybody loves an orchid. Many people don't know they're actually looking at an orchid, and I haven't met a single person who doesn't like an orchid. And it's been one of those things in my life that with my grandmother, we used to go orchid picking.
Picking?
Because the bush was inexhaustible. There were orchids everywhere, and we used to go looking for spider orchids. And I just was enamoured with orchids. And I think somehow there's orchid genes that put into humans at an early age, and you never get rid of them. And so one of my great joys was starting to think about doing research projects on orchids. And so when I started my PhD, I went to my PhD supervisor I'd chosen, very distinguished nitrogen physiologist, so worked in legumes, and he said, well, we've got a plan for your PhD. I said, I really want to do a PhD on orchid ecology. And he looked down his nose over the top of his glasses as he was wont, and he said, orchids grow too slowly and nobody knows much about them. I think you'll work on tuberous plants. And I agreed because I thought, well, orchids have tubers, so at least I'll get in through the back door. So we did some of the first ecological work on orchids, but from that day we built up a research lab, and of course it was well known at that stage that certain orchids used sex in their pollination system, so deluding poor dumb male wasps and hoverflies into believing that they're a wonderful female, but all they are is a hunk of vegetable that releases the sex fragrance that the male finds irresistible. And it's an extraordinary system. And Western Australian orchids, we've got about 400 species in the southwest. We have about 80 species that do it, and do it really well, with clear evidence that they're still beginning the process of speciating. So we're detecting new species embedded in an existing species that are experimenting with new fragrance chemistry to bring a different wasp species. And that's happening before our very eyes.
Oh, that is, and before our very noses.
Well, we can't smell it.
We can't.
No. If you can smell it, then you're a thinnered wasp male.
So the wasps don't get hurt in any way. They just get disappointed. They get fooled. A bit disappointed. I mean, I don't know.
Well,
I don't really understand wasps that well, but if I was a wasp, I would be a bit disappointed.
It's fascinating, of course. The orchid is better at producing the sex fragrance than the female wasp. So the orchid could actually dominate the reproductive cycle of the wasp and make it extinct, because this wasp, but it has an inbuilt sort of system where at about the 6th visit or 8th visit, that's the general thinking, the wasp learns and never visits the orchid again. Even though it's the most perfect chemistry, there's something that the orchid has put in there, some sort of very planned engineering obsolescence that says after a while you go, nah, it's like trying to repair that old broken clock. Nah, this is just never going to work. I'm never going to have those clocks again, and you'd never go towards it. So there's that sort of thing. That's something we really dream about resolving. What is the thing that they put in? Because they all do it, all those 80 species.
Well, if they didn't do it, the wasps would die out, wouldn't they? And then
In one generation, because the orchid's very effective.
That's amazing. The bulk of your work, I think, though, has been exploring the link between orchids and mycorrhizal fungi in the soil. So, can you tell me a little bit more about that? Because you have done a lot of work in that area, haven't you?
Yeah, and that goes right back to the second year of my university undergraduate degree, where I contacted a person called Jack Walkup. He was in South Australia. He's one of the first people in the world to be isolating, that means growing, the special fungus that all of our bushland orchids have in their root systems. And I wrote to him, and he sent me a little vial of the fungus of a greenhood orchid. And I remember it arriving in its test tube, and I thought this was magic. And I put my seed with it. Remember, this is a very young, green, budding botanist, and I got my first seedlings, and I thought this was like the most magical thing ever. Tragically, that had to wait 5 years until I finished my PhD, went to Kings Park, and so the first thing we did was we set up our first little trials. We started growing the orchids, and then the orchid program just grew topsy-turvy. Lots and lots of students. We probably had almost 20 PhD students and done heaps on mycorrhiza, worked with the Royal Botanic Gardens queue, saving rare orchids. And today we still continue our orchid program, but we're moving into new territory where we're taking the science and bringing it across to the community and empowering the community to do the sort of stuff that looks very white lab coaty and very so high tech, but in fact it's not. And you can get mums and dads and school kids doing their own little mycorrhizal programs. And our program that we're starting this year is training Indigenous kids through the b3aef to grow our native tree Cymbidium orchid, which tragically has been mostly removed illegally by collectors. People love seeing these orchids and rip them out of the trees. And so we're developing a method to train these kids to run their own mini lab without all the complicated technology to grow them and put them back along the Gibb River Road so we can all enjoy Cymbidium orchids hanging out of the trees when we do the great trip.
Well, that's a fantastic project. But how does a project like that get funded? How do you work it so that, great community science can be done?
Well, great support, and the Australian Orchid Foundation got a bequest for some research to be done on Cymbidiums, something on Cymbidium orchids. We all know what a Cymbidium orchid is. And I just thought, this is a project that I've just been dying to do. So I pitched it, and they said, this is better than perfect. And so it just got funded in the last 2 months.
Well, that's fantastic. I can't wait. I can't wait for it to happen, and I want to drive the road then, because it will just, that's for me. Look, going on an adventure, travelling, part of the best bit for me is driving along and seeing something and stopping and having a look.
Yeah.
When you're into plants, you're just stuck, aren't you?
That's right.
Everything has to be taken slower. Now, one of the work you're doing now is working about landscapes and the environments. One of the things I admire is you work with lots of people from different disciplines. So why? How has that worked? You've already given us one example with the engineer who made the machinery. In the past, I think botany was sort of done singularly, just botanists together. Why do you like that approach?
I guess if we go back to Joseph Banks, he was a botanist and first president of the Royal Society and all very eminent things, but he never uttered an animal name. He wouldn't have collected an animal. He would have seen the most extraordinary things, but he loved plants. He wrote about plants. His whole life was about that. And I guess working initially for a botanic garden means you generally do stuff on plants. But when we started with the whole journey in the mid-80s on restoration, how do you rebuild landscapes, it became very obvious that we were, as botanists, because we put the green stuff back together again, it was very clear that we were going to miss the mark big time, because what do we do with bird-pollinated plants? How will we attract them in? What about the insect pollination, which is a really big deal for us? What about the soil microbes, the ants that disperse the seed? And it started to be very obvious that we had to, God forbid, speak to a zoologist. And in fact, that was one of the first journeys that we made, was talking to the next door department that we never spoke to, which was the zoology department. We started working in the first one on native bees and how much restoration we would need to put in an area to support bee pollination. because without bee pollination, we wouldn't get seed set, so our restoration would fail. And from there, we've started working with a whole range of zoologists. Now we're working with a range of engineers, and now we've branched out to health professionals, because what we've discovered is restoring bushland, restoring forest is also a matter for many people of life and death in terms of becoming healthy individuals. I call it the indoor plant syndrome. Why do we have struggling little indoor plants? It's because that's a little connection to something that we find great peace with, which is plants, gardens, and the natural environment.
Wow. It must be really exciting having all these different groups coming together and doing things together.
Yep. It's challenging, but the great rewards are there. And I encourage all people in their disciplines to think outside of the box.
So what can you see happening in the next few more years you've got working in this area? What do you think?
Well, January was the beginning of the UN decade of restoring the planet, right? We've got 1 billion hectares to restore. It's an enormous undertaking. It's a great challenge, but we're up for the challenge. And I think nationally we can lead the world in showing that although we've degraded almost 48% of the land area of this continent in just 200 years, that we can rebuild it. And I hope I have a little piece of the science and the knowledge to help that journey and help it. So in 10 years' time, we should start seeing real Banksia woodlands, real wheat belt woodlands. We can walk through and go, wow, look at those orchids, look at those cottontails, leschenoltias, guinea flower, you name it, that they're all there and they're doing their thing, because a number of those things still are difficult for us.
Kingsley Dixon, it's been just a pleasure speaking with you today. I'm really more excited about the environment, hearing about all the struggles you've had, learning as you go. And I know that you're going to have lots more good ideas and discover lots of things to make the world a better place. So thank you very much.
It's a great pleasure. Thanks, Darren.